Welcome to this lesson, Cellular Respiration, one of the most fundamental and fascinating processes in all of biology. Whether you are a student preparing for examinations, an educator looking for a comprehensive resource, or simply someone that is curious about how your own body generates the energy that keeps you alive, this video is for you. My name is not important. What is important is what happens inside every single cell, every second of the day, without you being consciously aware of it. Right now, as you watch this video, approximately 37 trillion cells in your body are burning fuel. They are dismantling glucose molecules, passing electrons along molecular chains, and spinning microscopic biological motors, all to produce one tiny molecule, adenosine phosphate, or to call ATP. ATP is the energy currency of life. Without it, every process in your body, your heartbeat, your thoughts, the movement of your eyes across this screen would cease within seconds. In this video, I will take a deep journey through 4 major stages of cellular respiration. We will explore the structure of the mitochondrion, the organelle where most of this energy production takes place. We will dissect the glycolysis step by step. We will trace carbon atoms through the Krebs cycle. We will follow the electrons down to the electron transport chain, and we will examine what happens during energy production, during exercise, and how a poison like cyanide can shut the whole process down. This is not a surface level, far from that. This is advanced A-level biology. Stay with me, because the detail is where the real understanding lives. Let us begin. Before we dive into any single stage, let us look at the complete picture. The first stage journey that glucose takes from the cytoplasm all the way to being ATP. The first stage is glycolysis. This occurs in the cytosol, the fluid portion of the cytoplasm, and it does not require oxygen. Glucose, a 6-carbon sugar, is broken down into 2 molecules of a 3-carbon compound called pyruvate. The net yield is 2 ATP and 2 NADH molecules. The second stage is the link reaction, also called pyruvate oxidation. This occurs inside the mitochondrion, specifically in the mitochondrial matrix. Here, pyruvate is converted into a 2-carbon compound called acetyl coenzyme A, or acetyl-CoA. In the process, carbon dioxide is released and NADH is produced. This reaction requires oxygen, and it occurs twice for every glucose molecule. The third stage is the Krebs cycle, also known as the citric acid cycle. This also occurs in the matrix, where acetyl-CoA is fed into a circular series of 8 enzyme-catalyzed reactions per glucose molecule. Per glucose molecule, this produces 2 ATP, 6 NADH molecules, and 2 FADH2 molecules, and 4 molecules of carbon dioxide. The fourth stage is oxidative phosphorylation. This is powered by the electron transport chain, and it occurs across the inner mitochondrial membrane, and it is responsible for approximately 80 to 90% of the ATP that is produced. The electron carriers NADH and FADH2, how to remember them, in the earlier stages donate their electrons to this chain, driving the synthesis of approximately 28 to 32 additional ATP molecules. I want you to notice the locations. Glycolysis happens outside the mitochondrion, and every other thing happens inside the mitochondrion. This is why the mitochondrion is rightly called the powerhouse of the cell, and we are about to explore exactly why. The total ATP yield from one glucose molecule in aerobic conditions is approximately 30 to 32. I want you to compare that to just 2 ATP molecules from anaerobic conditions, and you can immediately appreciate why oxygen so critical to sustaining life as we know it. The mitochondria. Now, let us study the organelle that makes aerobic respiration possible. Look at the image on the left. This is a schematic cross-section of mitochondrion based on the transmission electron microscope, or what we call TEM images. When you insert your acto-10 I want you to spend some time looking at it. The mitochondrion has two membranes. The outer membrane, which is smooth and relatively permeable. It contains proteins called porins, which allow small molecules and ions to pass through freely. I want you to think of it as the outer wall of a factory. It defines the boundary, but it is not where the real work takes place. The inner membrane is where almost all the action occurs, and it is highly impermeable, meaning that while most molecules can enter, not very many can cross it without having a specific transport. This impermeability is absolutely critical, as we will see when we discuss chemiosmosis. The inner membrane is extensively folded into structures called cristae. Cristae are the inward folds of the inner membrane. These dramatically increase the surface area available for the electron transport chain and the ATP synthase molecules. Think of them as the shelves in a factory. The more shelves you have, the more machinery you can fit into it. A cell with high energy demands, like the heart muscle cell, has far more cristae than a cell with low energy demands, like a fat storage cell. Between the two membranes lies what we call the intermembranous space, what we are going to be calling the IMS. This narrow gap is where hydrogen ions are found. Now, we are going to consider two other slides. I want you to look at the structure diagram that has the liver cell mitochondria. And I want you to look at the structure diagram that has the cardiac muscle cell. And I want you to compare them. Notice that one has more cristae and the other has less cristae. When you look at the one on the left, which is from the liver tissue, I want you to notice that its cristae are relatively sparse. They're not very crowded. All that which is from the cardiac muscle on the right has dramatically a higher number of cristae. This shows you how structure and function come together without even us having to write it. The cardiac muscle, because it has to contract more, has more cristae because it needs more ATP. It has a large surface area for the electron transport chain complexes and more ATP synthase molecules compared to the liver cell. And I want you to think about this and think about other cells in your body and how they can vary when it comes to their ATP molecules. Let's talk about ATP. We need to fully understand the molecule that we're trying to produce, ATP synthase, but ATP adenosine triphosphate. I want you to look at its structure diagram. ATP consists of three parts: adenine, a nitrogen base, a 5-carbon ribose sugar, and 3 phosphate groups that are connected in a chain. It is the bonds between these phosphate groups, specifically the bond between the second and the third phosphate, that store energy in a chemically accessible form. The hydrolysis of ATP releases energy according to that equation. ATP plus water gives us ADP plus a phosphate group and 30.5 kilojoules of energy. This reaction is catalyzed by enzymes called ATPases, and the energy released is used to drive almost every energy requiring process in the cell. The energy powers muscle contraction via myosin ATPase. It drives active transport, for example, the sodium-potassium pump that maintains the nerve cell membrane potentials. It fuels biosynthesis, protein synthesis, nucleic acids, lipids, synthesis of those things. It enables nervous impulse transmission, among very many other processes. Here's a conceptual question. Why does the cell use ATP instead of just burning glucose? Think about it. The answer is precision. Glucose releases approximately 2,870 kilojoules per mole when it is fully oxidized. This energy is far too large to be channeled safely into specific cellular tasks. ATP, by contrast, releases a small, manageable packet of energy, 30.5 kilojoules. This is precisely matched to what individual enzymes and molecular motors need. I want you to think of ATP as a currency. You would not pay for a cup of tea with a 50,000 note and expect change. The cell uses ATP as the coin of our body, small denominations or small transactions. ATP can be regenerated from ADP and a phosphate group by three mechanisms. We have substrate level phosphorylation, I want you to keep that in mind, which we are going to see in glycolysis, the Krebs cycle, and we have oxidative phosphorylation, which we are going to find in the electron transport chain, and what we call photophosphorylation. This occurs in the chloroplasts during photosynthesis. You should know about photosynthesis by now. All three ultimately depend on the energy stored in the chemical bonds. They just access that energy in different ways. Glycolysis. Now, the first and the oldest metabolic pathway known to life. This metabolic pathway occurs in the cytosol of every living cell, and it requires no oxygen. Its conservation across all domains of life, from bacteria to humans, tells us that it evolved before oxygen even became available in the Earth's atmosphere. Glycolysis can be divided into two broad phases, what we call the investment phase and what we call the payoff phase. The investment phase, which is in red, covers steps 1 to 5, and the payoff phase covers steps 6 to 10. In the investment phase, the ATP molecules are consumed to activate the glucose molecule. Step 1, glucose has to be activated, which we are calling phosphorylated, by the enzyme hexokinase, using 1 ATP to form what we call glucose 6-phosphate. This traps glucose inside the cell because the charged phosphate group prevents it from diffusing out through the membrane. The second step is where the glucose 6-phosphate that we have just formed is isomerized to induce what we call phosphoglucose isomerase. It's isomerized by phosphoglucose isomerase to form what we call fructose 6-phosphate. This converts an aldose sugar into a ketose sugar. Remember our information on monosaccharides? Doing this prepares glucose for the next critical step, and this is very important. If you've been dozing, this is time to wake up. The third step is where we have phosphofructokinase, or what we're going to be calling PFK, so that I don't bite my tongue. PFK adds another phosphate group using another ATP molecule to form fructose 1,6-bisphosphate. This is the single most important regulatory step in glycolysis. Why? Because PFK is inhibited by high ATP concentrations. That means that when the cell has plenty of energy, glycolysis actually slows down. When ATP is low and adenosine monophosphate, which is AMP, is high, PFK is activated and glycolysis speeds up. This is an elegant and automatic feedback control. Now, pay attention to that step, because later we are going to be asking ourselves how glycolysis and the Krebs cycle are regulated. And this is one of the ways the glycolysis is regulated. The fourth step is aldolase. Aldolase, which is an enzyme, leaves the 6-carbon fructose 1,6-bisphosphate into 3 carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Step 5. Triose phosphate isomerase converts dihydroxyacetone phosphate into a second molecule of glyceraldehyde 3-phosphate. So, instead of having DHAP, we are going to have J3P. Pay attention to that. Now, we have 2 identical 3-carbon molecules and the payoff phase begins. So, the first phase, which is the investment phase, has given us 2 molecules of glyceraldehyde 3-phosphate. In step 6, J3P dehydrogenase oxidizes each glyceraldehyde 3-phosphate molecule, reducing NAD+ to NADH. This produces 2 NADH molecules. These are important electron carriers that will later donate these electrons to the electron transport chain. The 7th step is where the enzyme phosphoglycerate kinase catalyzes the first substrate level phosphorylation, transferring a phosphate group directly to ADP from 2 ATP molecules. We now have recovered the 2 ATP molecules that we have invested, that is in step 7. In step 9, steps 8 and 9 involve the repositioning of the phosphate group via phosphoglycerate mutase and dehydration by enzyme enolase to form the high energy molecule phosphoenopyruvate, or what we call it. Step 10. The enzyme pyruvate kinase performs the second substrate level phosphorylation, transferring the phosphate from phosphoenopyruvate, which we call PEP, to ADP, generating 2 molecules of ATP and producing 2 molecules of pyruvate. The net result of glycolysis is 1 glucose molecule, yielding 2 pyruvates and a net gain of 2 ATP molecules, 2 NADH molecules, and 2 water molecules. All of this with 0 oxygen required. To look at that. Phosphofructokinase is the pacemaker of glycolysis. It sits at the 3rd step of the pathway like a valve controlling a pipe, and its activity is determined entirely by the energy status of the cell. When ATP is abundant, it binds to an allosteric site of PFK, a site that is different from an active site. For those that are thinking about this is what we call a non-competitive inhibitor, and causes a conformational change that reduces the activity of PFK, which causes glycolysis to slow down. The cell is in fact saying, I have enough energy, so I want you to slow down production. However, when AMP is high, indicating that ATP has been heavily consumed, AMP instead binds to the PFK and activates it. Glycolysis accelerates, and in this case the cell is saying, I need energy, make sure you're producing a lot of it. Citrate, when it accumulates in the cytoplasm, also inhibits PFK. This is a signal that the Krebs cycle is running slower than glycolysis, and acetyl-CoA is backing up. Insulin and glucagon also modulate PFK activity, linking the glycolytic rate to whole body hormonal signals. This level of precise multi-input regulation ensures the cell never wastes energy, producing ATP that it does not need. Let's look at this diagram. This slide simply shows us a visual reference. I want you to be able to trace each intermediate from glucose all the way to pyruvate. Pause and think about it. Look at it. Zoom in if you have to. Now, when you are studying this diagram, keep your eye on several key molecules. Glucose 6-phosphate is the first intermediate. Once formed, glucose can no longer leave the cell. Fructose 1,6-biphosphate marks the point of no return in the investment phase. In other words, once that happens, glycolysis has started. Glycerol 3-phosphate, or what we call G3, is a 3-carbon molecule that enters the payoff phase. And remember, you get 2 of these from each glucose. I want you to watch for the moments where NAD+ becomes NADH at step 6. This is where electrons are harvested and stored for later use in the electron transport chain. And note that these 2 points, step 7 and 10, where substrate level phosphorylation directly produces ATP. The key molecule list on the right side of this slide summarizes all the intermediates and their full names. Use this as a reference when you review the detailed step table in your notes. Being able to name every intermediate in glycolysis, giving the enzyme and stating the product is an essential examination skill. Now, glycolysis is completely reversible except for 3 steps, those that are catalyzed by hexokinase, phosphofructokinase, and pyruvate kinase. These irreversible steps are the ones that are heavily regulated, which makes biological sense. You don't want your cell to accidentally run the pathway backwards when it needs to be producing energy. Let's talk about the link reaction, or what we call acetyl-CoA. Pyruvate has been produced in the cytoplasm now, but to enter the Krebs cycle, it must first be converted into acetyl-CoA, and it must travel into the mitochondrion to do so. The link reaction, also what we call pyruvate oxidation, occurs in the mitochondrial matrix and is catalyzed by a large multi-enzyme complex called pyruvate dehydrogenase, or what we call PDC, the pyruvate dehydrogenase complex. This reaction occurs in 3 steps. First, decarboxylation. The carboxyl group of pyruvate is removed as carbon dioxide. This is the first of 6 total carbon dioxide molecules that will be released a glucose molecule during aerobic respiration. Second is oxidation. The remaining 2 carbon fragments are oxidized, producing NADA plus NADH. And the third step is acetylation, where the 2 carbon acetyl groups are going to be joined to coenzyme A by a high energy ester bond, forming acetyl-CoA. Because glycolysis produces 2 pyruvate of glucose, the link reaction occurs twice, yielding 2 acetyl-CoA, 2 NADH, and 2 carbon dioxide of glucose. No ATP is produced directly. The energy is entirely captured as NADH. Now look at the diagram on the right. Acetyl-CoA is not just a product of carbohydrate metabolism. It is the convergence point for all the 3 major macromolecule carbohydrate categories. Carbohydrates enter via glycolysis and the link reaction. Fats, specifically fatty acids, are broken down by a process called beta oxidation, which occurs in the mitochondrial matrix and generates large amounts of acetyl-CoA, along with NADH and FADH. Proteins, after having their amino groups removed by deamination, have their carbon skeletons converted into various metabolic intermediates, many of which become acetyl-CoA. This is why acetyl-CoA is called the metabolic hub. Regardless of whether you eat bread, olive oil, or chicken for dinner, the same two carbon molecules, acetyl-CoA, is what ultimately enters the Krebs cycle. The Krebs cycle does not know and does not care where the acetyl-CoA came from. A critical point that is worth emphasizing: in animals, acetyl-CoA cannot be converted back into pyruvate. This means that fat which enters metabolism as acetyl-CoA cannot be converted back to glucose. This is why dietary fat cannot replenish blood glucose during fasting, unlike protein, which can contribute to carbon skeletons or gluconeogenesis. Let's talk about the Krebs cycle. We have arrived. This is also known as the citric acid cycle, the tricyclic acid cycle, named after Sir Hans Krebs, who elucidated it in 1937, and he even received the Nobel Prize for it in 1953. The cycle of the Golgi matrix and turns once for each acetyl-CoA that enters. Since we have acetyl-CoA per glucose, the cycle turns